שלמים שם. יש שם המון בלה בלה, יש שם המון פילוסופיה, דברים שלא קשורים אלינו ישירות. לא הייתי קורא פרקים שלמים, זה בטוח. נחכה עוד איזה 2 דקות נראה לכם? לא יודע. טוב, נראה לי ש... Okay, so last time we talked about regulatory mechanisms in the nucleus of the cell, then transport of the RNA molecule to the cytosol through the nuclear pore complex. And there are additional regulatory mechanisms that happen in the cytoplasm. And they are called cytoplasmatic mechanisms of post-transcriptional regulation. So we are still at the same line of post-transcriptional regulation, but we've reached the cytosol. Okay, and here we will meet small regulatory molecules, which are relatively small in size. They are transcribed from the genome of an organism in the length of about 70 nucleotides. And so they are called microRNAs. So these are genes. They are in the genome of humans, of plants, of a lot of organisms in the nature, but they do not code for proteins. They remain as RNA molecules, and they have a function in the cell, and the function is to suppress or repress temporarily, transiently, messenger RNAs of other protein-coding genes. And so this suppression is via base pairing, and we will see it in a second. But again, these are regulatory molecules. They remain as RNA molecules, which was weird for a long duration of time, and researchers tried to find their role. And today we know lots about their function and where they are in the cell, and lots of biologically relevant instances for diseases and other mechanisms in the cell. Okay, so when they are transcribed from the genome, they are, as I said, about 70 nucleotides long. So these are called the precursor microRNA. And right after that, they are cleaved by ribonuclease. So ribonuclease is an enzyme that cleaves an RNA molecule somewhere in the middle, and that's why the endo is here. If that's a ribonuclease, and specifically it's an endoribonuclease dicer. Anyway, just ribonuclease, it's a protein or an enzyme that cleaves RNA molecules. Okay, so specifically they are cleaved by the ribonuclease, dicer, and then the mature microRNA are produced, which are about 20 nucleotides long. Okay, so these are the active, or as I said, the mature microRNA. These molecules, in their turn, they base pair with messenger RNAs for protein coding genes. The messenger RNAs that are targeted by microRNAs are called the target messenger RNAs. And there's some unique feature, and it's not accidental. It has a meaning, or we think that it has specific function in biology, that the base pairing between the microRNA and their targets it does not have a precise or a perfect complementary, but there are some mismatches along the base pairing. And we think, well, again, researchers have several ideas for why it is so. One of the ideas is it will allow a much faster evolution of microRNA regulation that will give rise to much, just higher chances for a beneficial mutation. Let's leave it like this. And this mismatching that happens between a microRNA and its target is one of the things that distinguishes microRNA-mediated suppression and another very similar suppression that is generally called interfering RNA, usually performed by small interfering RNA, and that's the next class of molecules we will mention. So short interfering RNA that perform interfering RNA, the phenomenon that is called interfering RNA. This time the precursor is a double-stranded RNA, in opposed to the microRNA case where it was transcribed from the genome as a single-stranded RNA. The next slide will be a summarization of the differences between microRNAs and siRNAs. So in a minute it will be more clear. siRNA undergoes perfect match, in opposed to the microRNA that undergoes a match with some mismatches with their targets. And this time, again, in opposed to the microRNA case, it caused a cleavage, degradation of the RNA, of the messenger RNA molecules, and not, as was in the microRNA case, temporal transient suppression of the messenger RNA. So again, the next slide will summarize all the differences between these two molecules. Just one more word before this slide. So we have experimental tool that was influenced by the natural occurring phenomena of interfering RNA. So in the lab, the artificial method that uses or that implement this concept of interfering RNA is called antisense inhibition, where we just synthesize RNA molecules that base pair perfectly with some target messenger RNA, and we can transiently knock down a gene. Instead of knocking out a gene from a transgenic mouse, we might get to it in the next chapter, but it's a very tedious work, prepare an engineered knock-out mouse, but you can also knock down transiently a gene from the mouse, just injecting to it an antisense molecule. Yep.
So as it's short interfering, does that have a negative connotation in the eyes of researchers? Like micro is neutral, but interfering usually has a bad
Connotation.
connotation.
What do you mean in connotation? In what sense?
In how we view its function.
I don't think there's ethics involved.
You're talking about ethics. That's the case of the engineer of.
No, not necessarily. I mean, it could be, but if you say something's interfering with something, it's usually it's like the same way that researchers used to call it junk DNA.
I see.
They had no reason. They thought it was,
The interference is with the translation or the continuation of gene expression pathway along the gene expression pathway for a specific gene. So interfering here, the term here is just a functional. It's in its functional meaning. But if we had mentioned ethics, so you know, engineering novel genomes is very well. That have a bad connotation for many populations, mainly in the U.S. and so on. And this is kind of a bypass of all these very the own issue that because you don't change the genome here, you just yeah. Good morning. We are free now. You don't change the genome this time. You just influence the RNA level, so it has a much more acceptance in the eyes as well of the outside the academia. Okay, so antisense inhibition, and now we've mentioned these different features that microRNA and siRNA regulations have, and now this summarizes what really happens in these two regulations. Okay, so as I said, microRNA precursor is a single-stranded RNA. That's the way it's transcribed from the genome, though it has a specific. It's called a hairpin structure. What you look at here, in which it base pairs with itself to form a double-stranded region, but it's a double-stranded RNA. As a whole, it's just one strand of RNA molecule. So this, as I said, this secondary structure for RNA is called the hairpin. We've actually mentioned it when we talked about different structures in RNA. In small interfering RNA, well, they're not transcribed as double-stranded RNA, but right after they are transcribed, they found a complement strand, and they are in the form of a double-stranded RNA. So that's unique for siRNA. Both these classes, or both of these molecules, will be cleaved by the endoribonuclease dicer, which will cleave them somewhere in the middle, give rise to a double-stranded product. A short one, about 20 or 25 nucleotides long. Now the next step is that just one strand of this product, of this product, will be incorporated to what is called the RNA-induced interfering complex RISC, which will then guide the RNA molecule to complement or to its correct target, and then in the case of siRNA, you will have a perfect match, which will cause a cleavage of the messenger RNA. And the meaning is, of course, that the protein that this messenger RNA codes for would cease to be produced. That's what suppression at the RNA level means. And in the case of the microRNA, you have some mismatches along the base pairing region. And this will cause, at least that's what researchers think up until today, and this will cause temporal suppression on the RNA molecule. So usually the microRNA case is the one that the RNA molecules are not degraded, are not cleaved, but they are suppressed, either by pulling there to some distant, remote, unfunctional location in the cell, where they are not functional, or by just interacting with the machinery that's essential for their translation. So some mechanism represses that transition of this RNA molecule, its translation to a protein. But it's still thought to be a transient, a temporal repression in the case of microRNA. So that's what you really need to know about these two classes of molecules. Okay, here are just two examples of biological function of microRNA in neuronal systems. So that's a Cell paper that shows that the system here is the retina of Drosophila, light adaptation of the retina of Drosophila. So when you take this retina and put it in a dark environment and then switch it back to a light environment and back again to dark environment, lots of the microRNA population is altered. And specifically, they show here the temporal profile of one specific microRNA. So you see that its levels decreases and then increases back again when you put it back in a light environment. And these are additional examples for additional specific microRNAs. And these two last columns are just to show you that it's not a global effect on microRNA regulation, but just there are some microRNAs that are not altered by this paradigm, but still you can find many others that do. And they proceed in this article to give some functional meaning to this alteration in microRNA regulation. Okay, another example is again for Alzheimer's disease. Okay, so what researchers did in this study, they grow what we call primary culture neurons. You can harvest neuronal cells from a mouse embryo and grow it in a petri dish for a couple of weeks. Then they've added the artificial amyloid beta, the toxic, what is thought to be the toxic protein peptide in Alzheimer's disease. And I think a week after, they've checked or they did an assay that is called a microarray. That it's a method that allows us to detect the level of plenty, hundreds or thousands of molecules at the same time. So they've checked the levels of all 1,000 known microRNAs, and they've shown that the global effect is that of a downregulation of the microRNA population when you add the toxic amyloid beta. That's compatible. This is something that others have seen also, the global downregulation of microRNA regulation. The other groups have seen it, and also us, actually. And then you can do the following, which we're not going into the details of this procedure, but just as a concept, I think it's important to know it. So if you check a microRNA, you can computationally predict its target genes, just by looking for genes which have sequences which will complement with this, taking into account some mismatches, but which will complement with this microRNA. So microRNA X will have some predicted targets. So this could be gene A, B, and so on. Now what they did here, what you see in the table, is that they checked microRNA X, Y, Z. For each one, they gather a group of predicted target genes. This one has a group for itself, and this one the same. And then they can ask some sort of a combinatorial question that will assume that if there is a gene that is targeted by more than one microRNA that was deregulated, maybe even by all of them together, so it will be an important gene. For instance, the gene that stands in this overlap between these two domains. And okay, so now you look at the genes here, say gene 1, 2, 3, 4, and you can ask if these 4 genes play a role in the same known mechanism, known molecular pathway, known molecular cascade. And again, this is something you ask by using the hypergeometric test, and you ask if this group of genes is enriched with the genes that you know to play a role in a specific molecular cascade. And if you ask this question and give a p-value for each of these, for each of the known mechanisms in cells, the first one that jumps out, comes out of this analysis, the exon guidance, which makes sense in the sense that, well, in Alzheimer's disease, we know that some of the exon guidance, at least proteins that are involved in exon guidance, indeed affected. So that's, okay, to conclude, that might imply that some of what we see in Alzheimer's disease is due to dysregulation of microRNAs. That's what you can take from this research. Okay, was that clear? The sort of the concept of the analysis between Mir and their target. Hi. Okay, so okay. Okay, not just RNA molecules affect messenger RNAs at the cytosol, also some regulatory proteins. As a whole, a messenger RNA molecule will have the following principal structure of it will have a region that will be coded to a protein. So this is where this is the coding sequence, its start site and its end site. So this is the region, the green one is really the region that will become a protein, and it has an untranslated region, the UTR, the region that will not become a protein. Flanking, you have two flanking regions of this sort, one in the 5' and one in the 3' edges. Upstream to the 3' UTR, you find the poly-A tail, which you mentioned, and downstream to the 5' UTR, you have the cap. Okay, so proteins that regulate messenger RNA translation at the cytosol usually bind to the 3 UTR or to the 5 UTR. These are the regulatory regions of the messenger RNA. Okay, most RNA molecules are found in the cytosol with a very long poly-A tail, and that allows them to interact cooperatively bind what is called the PABPI protein that interacts via additional adaptor proteins first with the cap of the same messenger RNA, which stabilizes the whole structure of the messenger RNA and specifically stabilizes the cap. And again, BABPI interact with an adaptor protein to recruit the small ribosomal subunit of the ribosome. And that's the first, not the first, that's one of the first step of the initiation of translation. Okay, so this is the translationally active form of a messenger RNA. You have to have a long poly-A tail for these proteins to effectively bind, interact, and recruit the small ribosomal subunit, which is an essential step in translation. If you have a very short poly-A tail, which is indeed the case for some messenger RNAs, then you have a translationally dormant form, because PABPI would not bind and would not recruit the small ribosomal subunit. And also, I would add that the stabilization between the 3 and the 5' ends is much weaker than what we see here. Okay, and this is the translationally dormant state. The most classical, famous example for a cell that uses this mechanism of dormant RNA molecules that will be polyadenylated at the cytosol is the example of an ovum cell, tabeitzit. So in ovum cells, there are lots of messenger RNAs with very short poly-A tails. They are in a dormant state, so this cell kind of waits for something to happen, but in a very active standby state. And then upon fertilization, the poly-A tail of lots of these messenger RNA will be elongated very effectively, very rapidly, and all of a sudden lots of things would happen because these dormant messenger RNA will become active in a very short time. Okay, so that was, I mentioned the ovum cell, but synaptic strengthening or neurons, when they learn new tricks, are much alike ovum cell in the sense that also in them you find messenger RNA with very short poly-A tail, which will be polyadenylated, which will be elongated with the income, the arrival of some outside signaling, and which will cause several things to happen, among them synaptic strengthening. And as it was the case for RNA splicing, which I've mentioned is a mechanism that's still thought to be mainly or even solely in the nucleus, but some researchers claim to see it in the synapse. It's an interesting mechanism, but this one is it's much more established in science because it's a very it makes sense in the way that in some cases these are specific synapses along a dendritic tree that strengthen. So it can be a specific synapse and that will strengthen another synapse on the same dendritic tree. So you have to explain it by some mechanism that do not pass through the cell nucleus or through the cell soma, and this is one of them. So if you imagine that the postsynaptic site has lots of dormant messenger RNA in it, as other synapse B and C and so on, but only in this synapse something has happened that caused elongation of the poly-A tail for this messenger RNA, and then all of a sudden they are changed and they are translated, and lots of proteins are produced only in this synapse and give rise to a specific phenotype only for these synapse. So any explanation that explains specific changes in a subregion in the dendritic tree is something researchers are looking for. Polyadenylation is one of them. Sorry, cytoplasmatic elongation of the poly A tail is one of them. Give me a second. Okay, at the level of RNA molecule at the cytosol is a function of the rate of its synthesis down there in the nucleus, and also the rate of its degradation. And as it was the case for proteins, the degradation for RNA molecules is an active process. There are different processes in the cytosol that degrade RNA molecules. As a whole, we can say the different genes, meaning different RNA molecules, have different half-life or different lifespans. And an interesting phenomenon in this regard is that unicellular organisms have much shorter average half-life for their RNA molecules, about a tenth in length from multicellular organisms, and we think that it's because unicellular organisms are much more exposed to the changing conditions in the environment, and then they have to change their transcriptome, their population of RNA molecules and population of proteins much faster than multicellular organisms, which most of their cells are isolated from the environment because we are roped with skin and usually clothes. But okay, so unicellular are much more exposed and hence have a shorter half-life for the RNA molecules. The different mechanisms that exist for degradation of RNA molecules are the decapping pathway. You can degrade it from the cap side, from the poly A side, or somewhere in the middle. So begin with the decapping pathway, which we will not say much about, only that some endonucleases, again, a nuclease that cleave the RNA molecule somewhere in the middle, even if it's close to the edge, they can remove the cap, which will expose the RNA molecule to an exonucleatic decay, meaning a cleaving enzyme that will degrade the RNA molecule from the 5' to the 3' side, from the cap to the poly A side. So this diagram have the famous Pacmans in it. In next courses, you will see these Pacmans more often. We gave some examples where we saw the structure of what exactly this Pacman means. Here, I leave it in the level of the Pacman of the cartoon. But obviously, okay, so that's a protein with an active site, and you can go into it and see the atomic level mechanism of how it degrades the RNA molecule. We will not do it right now. Okay. For messenger RNAs at the cytoplasm, the poly A tail, its length is being decreased with time through the action of deadenylating nuclease. So the yellow Pacman continuously eats the poly A tail and shortens it. When the poly A tail reaches a certain length, first the stability between the 3' and the cap end, which was performed during the BPI proteins we've seen a couple of slides ago, cannot exist anymore, and the stability of this RNA molecule decreases very substantially, and in addition, it's now exposed from the poly A side to an exonucleatic decay, which is what the exosome performs. Okay, in parallel to that, the RNA molecules, when they are destabilized by the short poly A tail, in parallel to that, decapping can occur, which will give rise even to the 5' to 3' exonucleatic decay. So you can sometimes have an RNA molecule that's decay in parallel from both sides. Now, for RNA, they are degraded in the deadenylation-dependent pathway. There is a coupling. There's a coupling between transcription in the nucleus and degradation in the cytosol through the reciprocal interactions between initiation factors of transcription there in the nucleus and BABPI, which is the messenger RNA stabilizer in the cytosol. We will not go into the details here, but again, the faster this guy give rise to the following result, the faster or the higher an RNA molecule will be transcribed, the slower it will be degraded. We think we know the mechanism for why it is so, but again, if we look at mRNA level as a function of time, so for most RNA molecules, the higher the transcription in the nucleus, the slower the degradation. So if they have a slower transcription, it will have a faster degradation. Right? If this slope is steep, then this one is what's the opposite of steep? Matun, right there. Okay, so this coupling between these two processes occurs for most RNA molecules. Some RNA molecules, on the other hand, have to be transcribed very rapidly, but then also they have to be degraded very rapidly. Okay, they have to be also degraded rapidly, maybe because they play a part in something that happens in the cell and which have to give give room for the next step to occur. For instance, proteins that play a role in cell division, in mitosis. So these are very accurate in time proteins in their level of expression, and they are expressed and then neilamim disappears from the system very rapidly. And for these messenger RNAs, you don't see this coupling, and the thing that uncouple these two mechanisms is a specific sequence that they will have, which will first induce an endonucleatic cleavage in them, and which will then guide them to the exosome. So they are very actively, and maybe in a parallel pathway to the more canonical one, degraded. And so even if they have some reciprocal interaction between initiation factors and PABPI, you will not see it due to the sequence that calls for the exosome. If an RNA molecule has this sequence, it will be actively and rapidly degraded. Right. Okay, that's about it. I mean, this is where we end our discussion about this part of RNA metabolism. We will just mention two more facts. First, that the messenger RNA throughout his lifespan is under continuous surveillance. So if the RNA looks bad from some reason, it will be degraded. It will not become a protein. There are several mechanisms that perform this. And also, RNA molecules are localized in specific regions in the cell, and so they will be expressed in the proper place and proper time, which is again regulated very tightly in most cells. So that's it. We've seen the post-transcriptional modification in the nucleus, translocation to the cytosol, and some regulatory mechanisms in the cytosol at cytosolic level. Okay. I wanted to go through with you on the assignment. There are only 4, so let's wait for maybe some more students will come to the second hour. So for now, let's proceed with the next chapter. Okay, so this one is parallel relatively to chapter 9 in Lodish. And okay, it's called Molecular Genetic Techniques. Let's get into it via this slide. Okay, so when researchers want to investigate a gene, its function and so on, its structure, they use you can say tools from three different kinds. They use biochemical tools to determine the structure of a protein, its function, where it is localized in the cell, and so on. They also perform usually a genomic investigation, which asks if the gene has homologs in other species, and can you say something specific about its genomic structure. Then they would usually use some animal model or cellular model to see what task the gene performs in the whole organism, either by overexpressing the gene or by underexpressing it or by expressing some mutated form of the gene. So when we move from up to bottom along this diagram, this is what we call the classical genetic approach. Yes? From up to bottom, the green arrows, that represents the classical genetic approach, in which researchers identify a unique animal, some subject that has some specific morphology, some specific phenotype. And they next isolate the gene that's responsible for this outcome, for this phenotypic outcome. Then they can prepare recombinant DNA, which will allow us to prepare large amounts of the desired protein, which is, okay, you have to have large amounts of these proteins to biochemically investigate it, either crystallize it, or any biochemical assays. You have to have a lot of these molecules to be able to reliably say things about it. And then moving from bottom to up along this diagram represents the reverse genetic approach, which is, I think, more frequent today, that starts with the protein either because we know it has specific region motif in it. Maybe we know, it looks interesting at the level of its structure, or sometimes we even start here. We know that the gene is maybe unique for primates, or even it's unique to humans only. So we mutated it and inserted it into an animal model and see what it does. So the movement from here or from here up to the animal level is the reverse genetic approach. We will focus on certain points in this diagram in the following chapter. But we will start with some general terms in genetics. Yes?
Is it fair to say that it's active versus passive differentiation? That from the top down it's more passive, or you're observing what's already there, but from the bottom more experimental or active?
Yeah, I would agree, because you have to have, this is what Mandel did with his PIN study, and this is what, today we have so many methods to go up this diagram. I think most researchers, so you would say this is more passive and more active. Yeah, I guess, yeah, you can say that. I see what you mean. Okay, so some basic genetic terms. Not to put us all on the same line, but just to, the basics have to be before we go into the chapter itself. So allele, or alleles in plural, any population of organisms. So for a given gene in this population, there are several variants, naturally occurring variants for a given gene in a population of organisms. These are still not mutations, because they happen in nature. Mutations, we reserve this term for something else. So the different naturally occurring variations in a gene are called alleles. The genotype, when we say genotype, we mean the whole set of different alleles for the genes an individual has. So the set of alleles, that's the genotype, though researchers usually use it in the context of a study. A genotype is the gene under investigation or the group of genes under investigation. So that's what genotypes in articles usually mean. Phenotype is the physical attributes or traits. So this is as the genotype was the set of the alleles, so the phenotype is the set of traits, physical traits, though phenotype in neuroscience is also behavior sometimes, so don't be confused with the physical traits. Could be behaviors. Phenotype is the set of traits or behaviors an individual has, and we think that it's the outcome, the direct outcome of the genotype. And again, in articles, phenotype is usually means the specific trait or group of traits or behaviors that are under investigation. Okay, the naturally occurring, the most normal, the most frequent form of allele, which in this sense is normal, is called the wild type. So if one allele in a population is 99%, that will be the normal, the wild type allele. And we use the term mutation usually when we see a new allele being formed, either in evolution or in a study. We induce some, we give rise to some new allele. So that's a mutation. That's what researchers call a mutation. And a mutagen will be any agent, a substance or radiation or any agent that will cause a change in the DNA, which is obviously a heritable change because the DNA transforms to the next generation. All right? Okay, we'll talk a little bit about mutations in this chapter, about cloning a DNA, characterizing the cloned DNA, and different goals we can achieve with it. Different, we'll mention some usages for it. We will hopefully get to some words about genomics, which is a continuation of a way to use a cloned DNA, and we most usually will not get to this last point, because as I say, I want to go with you over the assignment, which deals with inactivation of genes in organisms. Okay, maybe let's do another slide and then take the break. Okay, so usually multicellular organisms are diploids. That means they have two copies of each allele, of each gene, two copies of each chromosome. That's like saying the exact same thing. Okay, now if a diploid organism, we are diploid organisms obviously, as you know. If a diploid organism carries two identical copies of the same gene, two identical alleles, allele X and allele X, so X is the name of the chromosome, so let's not confuse, allele A and allele A, it will be called homozygous for this allele. If it carries two unidentical alleles, say allele A and allele B, because you have two copies, it will be heterozygous for this allele. So homozygous and heterozygous are terms used for a given gene according to what's for the copies of alleles you have for it. Okay. In recessive mutations, or recessive mutation would be manifested at the level of the phenotype only when you would have two copies of the mutations, only when you would be homozygous for the mutation. Dominant mutations would be manifested at the phenotypic level, the organism level, even when you have just one copy of the mutant. It's sufficient to have one mutated copy to see some phenotypic effect. If that's the case, that's a dominant mutation. And that's the case where you are heterozygous for the mutation. Okay, so this table summarizes the different states for the genotype and phenotype in diploid organisms between the two different mutations. So if you have two normal, most frequently occurring alleles in your genotype, so you would have the wild type phenotype. If you have a single mutated copy and one of them wild type, one of them is a wild type, but the mutation is a dominant mutation, then it's sufficient for the phenotype to be that of the mutant form, the mutated form. If you have a single copy of the recessive mutation, on the other hand, it will not be sufficient, and you will still show the wild type phenotype. And if you have two copies of a recessive mutations, only then you would show the mutant phenotype. So that's what happened in diploids. In haploids, there is no sense talking about dominant and recessive mutations because it's enough for a single copy to show the phenotype. You know what? Let's just do one more slide because it's too direct to the last one. Okay. Usually, recessive mutations would be sorry, I lost myself. Would be a loss of function. Usually, recessive mutations would be a loss of function of a gene, of a protein. Because usually the case of loss of function would be compensated by the other normal wild-type copy. So if you have one defected copy, and you have one properly functioning copy of a gene, of a protein, that's enough for the function of the protein to be properly achieved, and you would have the wild-type phenotype. So this is, as I said, this is the case of loss of functions mutations. And usually dominant mutations are gain of functions mutations. That could be a gain of a toxic function, but that's still a gain of function. So if the defected copy caused the protein to be expressed in the wrong place or the wrong time, spatially or temporally, the wrong place or the wrong time, then it will gain a new function that was not in the original wild-type copy. So this is what we call a gain of function mutation. And in this case, it's intuitive to see that it's enough to have a single copy of the mutation for a bad phenotype to occur, or for the mutated phenotype. Usually it's a bad phenotype to occur. Okay, and we just have to mention that in some instances, though rare, dominant mutations can still lead to loss of function. When would that occur? When you have to have two copies of the wild-type allele for normal function. It's a dose issue. Because if you have two copies of a gene, you would have more RNA molecules, more proteins. And if you have these, you have to have a specific high levels of this protein for, I don't know, for proper lipid metabolism or anything. So you would see a change even with a single mutation, with a single copy mutation, and this is what we define as dominant mutation, that it's enough to see to have a single copy to see a change at the phenotypic level. Okay, so was that okay? Okay, I will not give any examples for recessive or dominant mutations. We gave some actually along the course. It's enough to open any journal that deals with what is called psychiatric genetics or molecular psychiatry. Well, you can see by the title, this is the most direct journal I know of that deals with exactly these issues, genes, brain and behavior. So that's exactly what we're talking about here, to see dozens of examples. Just as classical genetics try to attribute biological function to genes, specific genes, sometimes small groups of genes. So the same thing happened in neurogenetics or neuroscience, in which we try to attribute neuronal functions or behavior sometimes to specific genes. And you hear every once and then stories about specific gene that induces this behavior or another. Okay, so that's that about mutations. We will proceed with this one after the break. Let's take a 15-minute break. אם אתם רוצים פחות, אפשר גם פחות. אתם 4, כאילו, אתם יכולים לבחור מה שבא לכם. אתם רוצים להמשיך? אוקיי, אתם רוצים רגע הפסקה.
של הכל
כי
בזמן.
תביא את הנטייה לעבור אחרי זה שפרינצק, אני אעביר לך... זה לא תלוי בי, אבל לשכנע את מונה וכאילו...
אני אדבר איתה, אני
זה מישהי שהגיעה.
אני לא מכיר.
לא, מי שהגיע בשמונה, לא היה לי בעיות כנראה.
מה זאת אומרת?
אתה לא יצאת בחמש
אני
מהבית.
התעוררתי בשש בבוקר בשביל
גם
להגיע.
אני. ויצאתי, ולעבר אוטובוס, ואשפוז בלילה, המון זמן.
גרתי את חיי.
והתיק שלי רטוב, הכל רטוב, בפנים, רטוב. גם הנעליים.
לא הגיוני. אין לנו בעכו דברים כאלה.
בעצם מה יש בעכו?
קונטריינט, זה לא...
מבחינת המגדיר, זה משם רחוק.
מבחינה כללית, כאילו, מה יש בעכו?
מה יש בעכו?
לוד.
לא יודע מה חום.
כן, עיר עתיקה.
רחת וחום. פסטיבל פעם בשנה. אנחנו מנויים.
את האמת, תשמע, אני אגיד לך משהו. עכו יש לה באמת פוטנציאל להיות אחת הערים. כי יש שם הכל, כל מה שיש בתל אביב, כאילו חוץ מהעניין שזה במרכז.
חוץ ממה שיש בתל אביב, יש שם כל מה שיש.
מה אתה אומר?
לא, חוץ מהעניין שזה במרכז, כי יש לך חוף. ויש לך את העיר
גם
העתיקה, כמו יפו ו...
כנראה שלא.
פה יש יותר ריבוי, אתה מכיר? הוא ריבוי?
בטח איתה.
אז הוא באמצעות של ELSC.
באמת?
כן.
וואי.
טוב, כן. חלקים.
לא, אני לא מכיר את השמות, אבל אני יודע איפה זה, זה מסעדה מאוד
You know that what?
מדהים.
With the close veins?
הוא רק מודקים כזה, בקנדה,
מחר יש מעבדת ביום רביעי, נכון?
יש יום מחר ומחרתיים. אני לא נשמע.
אבל אתה לא מתרגל את זה?
אני כן, אבל יש יום מחר ומחרתיים. כאילו, כן.
זה מה זה מוזר המעבדות האלה? שזה על המחשב, כאילו, אין קשר בין זה, כאילו זה נראה
דווקא, במובן מסוים, אבל במובן אחר זה דווקא אחלה. תחשוב שמעבדת רטובות אתה 80% מתעסק עם המערכת העובדת, המערכת העובדת, אנחנו באמת צריך להניק שזה יותר, כאילו, זה לא באמת בסופו. תשמע, אני ביולוג, כאילו, בתפיסה, אתה בא לי, זה אחד, שתיים, למה לעשות ניסיון במאה החיים? בשביל מה?
לא חשבתי את זה.
גם אם מספרים לך שהבעלי חיים גם ככה השתמשו בניסוי אחר, זה רק בשביל
לנחם אותך?
לא, בשביל האתיקה, אתה לא אמור...
אתה היית במעבדת אתיקה, בטוח היית.
בסדר, אבל זה שימוש בעלי חיים, זה בסדר, זה כי אתה רוצה לדעת לעשות מחקר, ששואל שאלות שלא שאלו לפני 50 שנה, לא כמו בתואר ראשון, אתה לא מדביק זרם וצפרדע. זה כבר יודעים את התשובה, אז זה בסדר, זה אין לי בעיה.
באמת, איך הבעיה עם הקטע שנניח לוקחים לאדם מהעין או דברים כאלה במעבדה?
עוד פעם, אתה לומד שיטות שבשביל מחקר, בסדר, יש מחקר ויש לימוד.
כן, אבל אני מניח שגם המרצה...
יש לי בעיה, מספיק אין לי בעיה כדי שאני אעשה את זה.
כן, אבל גם המרצה שם בקורס הוא אמר שנניח את הדם שלוקחים להם מהעין או דברים כאלה, זה כל כך נדיר.
יש כאלה שלוקחים מהעין דווקא.
לא, השאלה היא אם אפשר ללמוד את זה במעבדה כשאתה כבר תקרא.
בסדר, נכון, אבל הם הגוף שנותן את האוטוריטה, כן, הם הגוף האוטוריטה, איך אומרים? אז הם אפשר לעשות את הקורס, זה כזה עניין...
אני יודע שפעם...
זה לפחות פעם אחת יהיה וטרינר עושה את זה, אין ספק שעושה את זה יותר טוב מהדוקטורנטית אצלך במעבדה שאני לך. אבל תשמע, בכל זאת וטרינר, כאילו... אנחנו שונאים אותם, הם מפריעים לנו בעבודה שלנו, אבל אם ככה, אז הם עושים את העבודה שלנו, בסדר? כאילו, קצת כבוד ללימודים שלהם.
שמעתי שהיה 10 מעבדות פעם בקורס הזה. 14.
באתיקה?
לא, בפסיכולוגיה. כן, כל הזמן הם אומרים שפעם, כאילו עד ממש, עד לפני 5.
לא, אני עשיתי את הקורס לפני 4 שנים נראה לי כבר.
אוקיי, זה לא היה איתו?
לא. אתה יודע, הגנות חובניות.
לא, זה אמרתי.
אצלנו חבר'ה התחילו לא לבוא,
I mean, I was learning a lot more from them. That's like natural way. It's just
שזה לא יפגע לך בתואר,
yeah.
ושמי שלא אפית לא מגיע למעבדה לטובה, לא יודע, יצטנף לכתיבת הדוח או בסדר, זה לא יפגע לו בציון. כי לפני זה מרצים אמרו כאילו,
מה זאת אומרת?
הביולוג זה תבוא לסוף הקריירה, אתה יודע,
אני אוכל להגיש לו איפה כאלה שמרשים את הטלפון?
כאילו, לא,
מאיר?
היום זה
אי אפשר.
כי היום יש לך גם הרבה אנשים שהם נניח משאבים פיזיקה וביולוגיה והם רוצים להיות כאילו מודליסטים או...
לא חשוב, לא בקטע אתי. יש דבר אחד אם לצאת נגד ניסויים בעלי חיים שהוא מחקר. לצאת נגד ניסויים בעלי חיים שהוא דבר ראשון. זה אני יכול לעבוד. כאילו מה זה? הדינוזאור, כן, כל ה... קבצ'ניק, כאילו דינוזאורים, ביולוג לא יכול... זה לא ככה. לא, זה מי שהם מחליטים ביולוג, מה הם מבדילים אם זה ביולוג או עליי? ביולוג לא יכול שלא פעם אחת לחטוף לו בצעדה. לא יכול להיות. גם שוב, מי בכלל? כאילו, אני לא מצטער, אבל... מה הם קובעים? לא יודע, ביולוג במדע? אז הם יגידו לך לפחות פעם אחת שמי שמנהל את הדיון תדע מה זה עפר של... הוא יודע, זה העול שלו איתה, מה זה חושב בכלל? בכלל זה המדען הוא ככה, המדע הוא יוניברסיטי. זה לא החלטה שלהם בכלל. Okay, so we've passed the critical mass to talk about the assignment. We will do it right after this, right after the following point. But still we are in, well, analysis of mutations, genetic mutations, and there is one elegant use of a double mutation system that I like to present. So it enables researchers to determine the order and the sort of interactions between components of a molecular cascade, of molecular mechanisms. So we've made a couple of such cascades, for instance, the division cascade. So how do researchers have determined that protein A is upstream in the cascade to protein B, and how did they determine if it's a suppressor, or an activator to some endpoint parameter, and so on. So one way to determine these, or to answer these questions, is the following. Let's assume that we know that mutation in gene A gives repressed reporter expression. Well, let's put things in our context. So let's assume that mutations in A gives repressed or lower levels of cyclic AMP. Right. Let's assume that mutation in the gene B give what is called constitutive expression, or give constitutively high levels of cyclic AMP. So we know these two facts. And now we perform a double mutation experiment, and there are two possible scenarios, two possible outcomes for this experiment. And for each one, there's only one explanation for the order and the interaction between these two components. So if upon a double mutation experiment we get a repressed or low levels of cyclic AMP, so it was the A effect that is dominant, that's affected this outcome. That means that A that is more directly linked to the reporter is downstream to B, and the only possibility that will explain this result and these two evidence is that B will be an inhibitor on A, that will be an activator on the reporter. So you can think about it more at home, but that's the only scenario that will explain these three results. Each time we have to explain three results. This one, the double mutation result, and each of the single mutation experiment results. And the second scenario is that we will get a constitutive high levels of cyclic AMP, and that means that it is the effect of B that was manifested in this outcome. And the only possibility is that A inhibits B, which inhibits the reporter, directly cause lower levels of cyclic AMP. This is one way to delineate, the interaction between components in a cascade. Yes.
I'm not entirely sure what a double mutant is. Is that when you just mutate?
Both of them at the same time, in the same system.
But you don't know what order they're in.
Well, we don't know. The order is not for the mutation. These are the thing is that these two genes play a role in the same pathway, and one acts upon the other, which acts upon another, a third component, and so on. So this is what I meant by the order of the components in the cascade. Double mutation is just mutate two genes together at once, at the same gene, at the same cell, same organism. So a reporter is anything, a gene, an RNA molecule, a protein molecule that reports or that we are interested in its levels. It can be the levels of cyclic AMP in the cell. I agree that here the reporter is something that is transcribed from the DNA. You see the double strand. So it can be a gene that's expressed or that does not express, and that will be the outcome that you will be after in these experiments. So I just tried to link it to what we have learned in the cyclic AMP cascade, for instance. So in this case, the reporter is not a gene from the genome, but some molecule, and its levels will be the readout of these experiments. So it's just a generic term for anything that will report an outcome of an experiment, I would say.
Yeah,
Yep,
how do
sorry.
you know they're not part of different mechanisms that affect the reporter?
Okay, I agree. Well, you have to it's kind of it's a framing question. Well, a single mutation in A and separately a single mutation in B affect the same something, and we know that this something. We have good reasons to think that there's a single mechanism. Well, you have to have more evidence or more data to to frame it in a single cascade. So one of the assumptions here is that A and B indeed play a role in the same cascade, and all you try to do is to order them correctly, and give minus or plus for the interactions. Okay, so that was a bit about mutation, analysis of mutation in neuroscience and actually in biology in general. Let's see, pretty briefly, how we clone a DNA. Okay, so DNA cloning or preparing a DNA clone enables researchers to prepare large amounts of identical DNA molecules, and as I said at the beginning, these amounts are essential. They are needed to certain biochemical assays. So you have to. That's a requirement when you start a biochemical study on a molecule. Usually you have to clone it in order to produce large enough amounts from it. And the term recombinant DNA is any DNA that is composed of DNA sequences, DNA fragments that come from different biological sources, meaning different animals. So that would be a recombinant DNA. And there's a wide variety of methods for DNA recombination preparation. Along the years, laboratories have developed very different and a wide variety of such methods. But as a principle, all of these methods follow these following principal steps, in which the first step will be that you link the DNA fragments. This is the DNA sequence we want to clone. We want to create a large amount of identical copies from it. You link it with a vector, which is another DNA fragment, another DNA molecule. And we call it a vector because it enables us to deliver the DNA of interest into a host cell. So even in drugs context, the term vector substance or anything, it's something that helps you deliver something else into a system, into a living cell, for instance. And this will give rise to recombinant DNA, because you'll see that most of the time a vector is a sort of a virus, a sort of a plasmid, and you link it with a DNA from a different animal, from a different source, which will give rise to recombinant DNA. And then we allow replication of this recombinant DNA within host cells, usually a bacterial cell system, following by isolation and characterization in many different ways of the cloned DNA fragment. That enables you to deliver the DNA of interest, the target DNA, in our case, to a living cell, insert it to the genome, or to incorporate it together with the genome of a living cell.
Why isn't it enough to just put the DNA onto the cell?
You can add just the DNA fragment to, for instance, bacterial cell cultures, and some bacteria would take the DNA fragment in, and even some of them would incorporate it into the genome. It's a much less effective process, and the vector has some special features in it that makes it an active infection. It's sort of the DNA fragment is infected into the living cell, and that's, it's a much more effective process, and it also gives rise to a stable cell line. So let's see the particulars of one method that prepare recombinant DNA, and maybe this question will be more clearly more clear.
Sometimes they use viruses to inject some DNA into cells and make it. So
Different vectors exist. Some of them are derived from viruses, HIV, FIV, and different sorts. If they use HIV, because I saw your eyebrows rise. So if they use HIV, they use the weakened recombinant HIV that has very little infective ability. What is called, there's a term for everything I just said. The last lesson, there's just one word for it. I can't recall. The beginning of the sentence, yeah. So let's see some particulars. In a minute, you'll see maybe this diagram will answer.
Happens replicate against the vector and give
Right.
one cell.
Let's go into a couple of slides, and if I still wouldn't answer your questions, so ask me again. Okay, so a vector can be many different molecules. Okay, in a minute a bit more about the vector, but before that, okay, so as one of the major objectives in DNA cloning is to obtain or to isolate a specific genomic region. Can be a gene, but can be an intragenic area also. And there are two components we need to mention that allow us to do that. The first are enzymes that called restriction enzymes, sort of molecular scissors that cleave the DNA in specific, well-defined sequences, and DNA ligases, molecular cellotapes that ligate, promotes the formation of the phosphoester bond between nucleotides in the DNA. A reminder, the DNA is built of nucleotides, so ligases promote the linkages between these building blocks. A bit more about restriction enzymes. These are endonucleases. They cleave somewhere in the middle of the RNA molecule. They typically recognize the specific about 4 to 8 sequences on the DNA. Typically, what is called a palindrome sequences, which are read the same from both sides, from opposite sides. So GAA-TTC, GAA-TTC. Most of them would give rise to these products, which have what is called a sticky end, meaning that single strand sticking out of the double strand structure. So it will be sticky because when we will add it to another DNA molecule, it will stick to it if it will be complement to this sequence. And when you take a given genome of an organism, and you would add to it, you would expose it to the same set of restriction enzymes or to a specific restriction enzyme, well, this genome would have the same set of restriction sequences upon it, and so it will give rise to reproducible outputs, reproducible products. It will give rise to the same products each time you do the experiment, or each laboratory that performed this same experiment will give rise to the same products. It will cleave the DNA on the same sites, obviously.
What
Sorry.
does the symmetry serve here?
It's a feature of restriction enzymes. It's something to do with their structures. I'm not sure actually, but it's not. We do use it in a way, but it's just something that comes out of their structure. It's just they are symmetrical along the point of cleavage. I'm not sure why it is so. Sorry? Yeah. So restriction enzymes cut in the following manner. So you get one product of this AATT exposed to base pair another DNA molecule, if you will, add it to the same test tube. So this exposed single strand is called a sticky end. Okay, so here we have genomic DNA fragments. You took a genome of an organism, you cleaved it with specific set of restriction enzymes, and you get these products. And this is the sticky end of the vector DNA. And if you put these two in the same test tube, well, only the DNA fragment that will complement, that will base pair with the sticky end of the vector, will link to it. So this would be a transient interaction between these two molecules. And upon addition of ligase, specific type of ligase to the system, it will promote with the formation of this phosphoester bond right here, which will just make this DNA again a continuous molecule, and which will give rise to a recombinant DNA. So when you look at that, you would not know that one fraction came from I don't know, fly, and the other from HIV. Yes, just one sec. Yeah, I think that was it, yeah.
These two groups, were they added, or does it just mean that we know which the phosphate group and the hydroxy group?
Yeah, if they are naturally occurring.
So it was just to show the action.
If they were added in a previous step. Well, nucleotides between them have a phosphate right here. So this would be the endpoint phosphate, and this one is the 3' end, so it has a hydroxyl. I don't think you have to add it artificially.
It was there, they didn't show it just in the previous.
The previous. Yeah, they didn't show it here, but obviously it's here. They are shown here to show that these are this is the phosphoester bond that's linked by the ligase, that its linkage is promoted by the ligase. Yes?
So is this also illustrating that the same vector can be used for different fragments?
That's absolutely true. The same vector can be used for different fragments. And you will see that a vector, would that be the next slide? Yes. So a vector would have a region with lots of different restriction sequences to allow a more versatile work in the lab. So you could use the same vector, as you said, for A or for B or for sequences from different organisms. But that was a spoiler for next slide. Okay, so now, and that's, yeah, the two last slides before we talk about the assignment would, so as I said, I mean, along the years, researchers have developed so many methods for recombinant DNA preparation, and we have so many different vectors for these methods. And maybe the most common one, still, I think, is the use of a plasmid. A plasmid is a circular double-stranded DNA that's naturally occurring. You can find it in nature, in bacteria. So it's a parasite. It's in bacteria. It's a virus in the sense that it has only a DNA. It has nothing else. It does not have its own proteins and so on. And it's a parasite because it uses the DNA replication machinery of the host cell to replicate its own DNA. So that's why we call it a parasite. Okay, so it was isolated, it's there in nature, but we have taken it out from nature and changed it in so many ways. And now we have many different plasmids that we use, but I would say that all of them would have the following three principal regions. The first, the open reading, what's the I4? I'm not sure. Initiation, I think, Well, the first point will be the sequence, a sequence where the DNA replication of the host cell recognizes and starts to replicate the DNA. That's the replication initiation point. You have to have that in order to replicate the genome, the DNA. A second region will be of a selection gene. And that means some gene that we will be able to select by the cells that have taken up these plasmids from cells that have not taken up these plasmids. And the next slide will show you what do I mean by cells that have taken up and cells that did not take up these plasmids. The most common use is a resistance gene to some kind of an give me 2 seconds. Ampicillin is a kind of antibiotic. Sorry, okay. So a type. So the selection marker would be usually a resistant gene to any kind of to specific kind of antibiotics. So here you see resistance to ampicillin. That's a gene that allows the cell that has this plasmid in it to whatever hydrolyze the antibiotics and so it's not effective anymore. So if you add this selective, if you add ampicillin for cells that have taken up these plasmids, they would survive, and cells that are not resistant to these antibiotics will not survive. This will give space, give more room for cells that have survived to grow in the tissue culture, for instance, and you will get and only cells that have this plasmid will proliferate. Again, the next slide will clear it, I think, more accurately.
Or natural design?
Well, some viruses has some mechanisms to fight our drugs as a whole, but still, yeah, that's something we have synthesized into the plasmid. That's part of a recombinant fragment. And the third region that the plasmid would have is the region where we will insert the DNA, exogenous DNA, the DNA of interest, the one that we want to clone. And this region it's synthesized, it's artificially designed to be rich with restriction sequences. These are names of different restriction enzymes. As I answered before, to give you a more versatile work in the lab, so you would be able to work with this plasmid, will give rise to one sequence of sticky ends, or with another restriction enzyme, which will give rise to another sequence of sticky ends. So these are the three principal regions of a plasmid: replication initiation point, selection marker, and the region to where we are going to insert the exogenous DNA, which is rich with restriction sequences. Okay, the sequence of events of what I've just described, just to go through it again using this slide. Okay, so we started with an isolated DNA fragment. That's the DNA fragment we want to clone. We want to produce lots of identical copies from it. You link it with a vector plasmid. We've just seen its principal structure. We get a recombinant plasmid. Yeah, you open this region. You open this region in the plasmid using some restriction enzyme, and you did the same for the DNA fragment, and you link them together with the ligase, and you get this product. Now you add it to a bacterial culture, and now some cells. These are the bacterial cells. Some cells will take up the plasmid and would hence be resistant to ampicillin. Because resistance to ampicillin, that's a feature that the plasmid has. Some cells will not be as resistant to ampicillin, and when you would add ampicillin to the system, only these kind of cells will survive. And that would give more, room in the petri dish for these cells to proliferate, while this one will perish, and you will get a clone. Okay, now because bacterial cells replicate in a mitotic manner in which each cell gives rise to an identical copy of itself, so you get a clone, and one more thing, and because the plasmid upon replication is being transformed to the daughter cells, and as I say, the daughter cells are identical to one another, you get a clone. You get a population of cells that each of them has the same specific DNA fragment the DNA of interest, the one we've inserted in the host cell. And I think it will be worthwhile to watch a short clip from the Lodis site of this sequence of events, which will, I think, clarify even further this sequence of events. And we will then talk about the assignment. So I had it prepared right here. Yeah, plasmid cloning.
DNA cloning is a method for isolating a particular sequence of DNA from a complex mixture of different DNA sequences. In order to clone a fragment of DNA, we need to insert it into a vector, usually a highly modified phage or plasmid that
Phage
can
is
replicate
is
in a
another kind
host
of
cell. Generally, a plasmid vector contains three elements: a cloning site where the foreign DNA fragment can be inserted, a drug resistance gene which destroys antibiotics, in this case ampicillin, to allow selective growth of the host cell, and a replication origin to allow the plasmid to replicate in the host cell. To insert foreign DNA, we must first use a restriction enzyme to cleave the vector at the cloning site. Here, EcoR1 cleaves the palindromic sequence
Specific kind of restriction
GAATTC
enzyme,
to
Eco.
produce single-stranded ends called sticky ends. These can hybridize with any piece of DNA that has also been cut with EcoR1. Foreign DNA containing the sequence we wish to clone is digested with EcoR1 and then mixed with the cleaved vector. The sticky ends of the foreign and plasmid DNA molecules hybridize and then are sealed into phosphodiester linkages by the enzyme DNA ligase, creating a recombinant plasmid. Each of these recombinant plasmids contains the inserted DNA fragment, an ampicillin resistance gene, and a replication origin. A pool or library of circular recombinant plasmids is thus created. Each plasmid carries a unique fragment of foreign DNA, as indicated by the colored arrows. Next, the host cells, E. coli bacteria, are added to the recombinant plasmids. The cells have been treated with calcium chloride to make them permeable to DNA molecules. Through a process called transformation, a few cells take up a recombinant plasmid, while most other cells do not. The bacterial cells are poured onto a plate of nutrient agar containing the antibiotic ampicillin. With ampicillin in the agar, only cells resistant to the drug can grow. At 37 degrees, the cells will grow and multiply. Because they cannot move on the agar, each will produce a separate colony of cells. The non-transformed cells lack the plasmid with the ampicillin resistance gene and die. Cells that have a plasmid are resistant to ampicillin and survive. The replication origin allows the plasmid to replicate by using the host cell's enzymes. Plasmid replication is independent of host cell division, but plasmids are distributed to each daughter cell when the host cell divides. As the plasmids replicate and the host cells multiply, the number of copies of the recombinant plasmid is greatly amplified. The multiple daughter cells form a colony or clone. Because all the host cells in a colony are derived from a single cell, they all contain copies of the same recombinant plasmid with its fragment of foreign DNA. A variety of assay methods can now be used on the bacterial colonies to determine which contains the particular DNA sequence we wish to isolate.
And you can even isolate a particular colony, just leave it with a tip and grow it on a separate Petri dish, and then you will have a clone of just one fragment of DNA. And how do you know that this is the right clone to take up? These are different. There are different answers for this question. And there were some additional details, as the fact that the cells have to be grown on an agar that will, they will be localized to a specific region, and hence the colonies will not mix with one another, and so on. So that's the basic of plasmid cloning. Any questions? Yeah.
How is the resistance? How are they resistant to the ampicillin? Is this the protein that is expressed by the gene?
It's a protein that expressed by this gene, and I'm not sure how exactly what it does to the ampicillin. Yeah, it does something to the ampicillin.
Okay.
Maybe it's if the ampicillin have to go into the cell with the receptor, so maybe it damaged the receptor. I'm not sure how it does it. I don't know. Yep. Second one.
I'm not sure.
I think it hydrolyzes. That's what I remember also. The ampicillin gene, resistant gene, I think hydrolyzes the antibiotic itself. Yeah.
Okay. And it says that the bacterial cell clones the plasmid, but it's unrelated to the mitosis.
Yeah, well, mechanically it's not, but it's not important to us. It's independent. That's correct. It's independent to the replication of the cell, but still you would have statistically distribution of the plasmids in daughter cells.
Yeah.
Just because the cytosol of daughter cells during mitosis is being split to two regions, and then the plasmid will be here and there, and
but the process of replicating
yeah.
DNA molecules with
Because in
mitosis,
mitosa, it's time coupled with with lots of other mechanisms that happen in bacterial cell, and the way that the plasmid DNA is cynically using the machinery of replication just makes it I'm not sure about the mechanism, just make it independent of the process of the endogenous replication that happens in the bacterial cell.
But what is its process of replicating DNA molecules that is not related to mitosis? Did we learn about such a process?
I'm not sure. I know what I understand the question. So again, in bacteria, the replication mechanism is coupled with other mechanisms. Replicating the genome of the bacteria happens in a specific lifetime of the bacteria, and certain mechanisms have to be mature enough biochemically for this process to happen, and so on. But the plasmid is independent of these processes. It just if it is recognized by the replication machinery, that it will be replicated, independently. We didn't get into it. It's not that essential. The basics is what we're after here, and I think we have it. Okay, just one word about next class. It will be okay. So we talked about analysis of mutation and DNA cloning. We will just the first hour of the next class, which is the class before the exam. We will give some instances of what do we do with the DNA, with the clone DNA, and what a huge revolution it did in biology. It gave rise to the field of genomics and so on. We'll just mention a couple of words about that. And the second hour, or the last hour and a half, I believe, for next time would be for us to answer together the huge question collection I sent you a week ago, I think. So try to answer it yourself. It will be much more effective for you in the class. If you know the questions already. Because we will do it quite briefly, because there are 26 questions, I think, so, and it will be good to go over all of them. And, okay, so I have here also the files I've sent you. Anyone want a hard copy for the assignment? So I printed a couple. So there's only, I think, 8. There's enough, okay. You can press it back, please. So the assignment is 40% from the final grade, and it tries. The idea here is, well, during the course we learn about terms in molecular neuroscience and mechanisms and so on, and also the papers we read. we read together tried to use these terms and to show how they are essential to understand, the function of an MDA receptor, for instance, and so on. So the idea here is to do the same thing only on your own and to, be convinced that indeed the course contributed to you something that you are now able to read, a hardcore biology article and understand most of it. Well, not all of it, but it's most of it. General instructions. So the submission will be in 5 weeks from now, the 14th of February. Okay, somewhere in the morning I'll be here around 9 a.m. and I will take it. Please prepare a hard copy of. Don't send it to me by email, just because I want to read it blank. And so also, as it is written, don't put your student's name on it. Just put the ID number or passport number. Well, just anything that I could identify you by. Okay, about the paper. So as you see, after choosing a paper and before starting the work, obviously before you start to work on it, please send the paper for me for an approval. And if you worry that I will recognize the work by the student that sent me the paper a month ago, don't worry, I will not remember who sent me what paper. So that's not a problem. But just the approval step is just for your benefit. It will be more. I would just suggest maybe choose another article if needed. So it will be more targeted, more directly linked to the questions in this essay. That's the idea. Okay, choose a paper. I'm reading from choosing a paper. Choose a paper that discusses a link between a molecular element and a neuronal element. What do I mean by a molecular and neuronal element? So a molecular element can be any cellular process, such as RNA splicing, protein folding and transport, any aspect, any mechanism we mentioned along the course, or as much as I'm concerned, even something we haven't mentioned. In addition, molecular element can be just a gene or a specific molecule, such as microRNA, which is linked with a neuronal phenomenon. It can just be a gene. You can choose a paper that delineate the function of, I don't know, SMU1, which is a gene in Drosophila neuronal system. Include genes for proteins that are involved in signal, etc. Neuronal element can be any morphological or functional aspect at the level of a single synapse, at the level of links, interactions between neurons, and at the level of the intact animal, behavior, for instance. The paper does not have to include details about the molecular processes of the gene. If you just mention a gene and then jumps right up to the behavioral level, then do some outside reading outside the article to know a bit about this gene and know what we know about it, and if it plays a role in some cascade, you should be aware of this cascade and so on. But you can choose a paper that focuses on either of the single synapse level, neuronal interactions, local circuits, or the whole brain, or the outcome, behaviors. Okay, and the specific questions are the following. Any questions about which paper is good for this assignment? Yes?
Do you have any recommendations on articles, on journals to look through?
I don't think, well, no, I don't have specific suggestions in this regard, though, well, just one comment that, for instance, Science and, huge impact factor journals like Science, well, not all of them, but Science anyway, is the articles are extremely short, about a page and a half, maybe 2 pages of article. And so in this case, you would most probably have to do some outside reading, because, it jumps right into the results sometimes, and you would have to have a background outside the article. I would assume that you would not choose a paper 15 pages long and so on, even though there are some. No specific journals that I can think of. Go with what you are more interested in. It's for your own amusement, this assignment. So, molecular psychiatry, immunity, current biology, cell, natural neuroscience, Journal of Neuroscience, I don't know. Okay, discuss the molecular element from the paper. If the paper discusses a molecular process, describe the process and its function. if the paper discusses a single gene or molecule, describe what is already known about the molecule. This should not be more than 200 words, this section. There are all in all 3 sections. The second one is to discuss separate, so the first one we discussed separately the molecular element. The second one we explain what is the readout, the phenotypic, behavioral or neuronal readout that the paper deals with. Discuss the neuronal element, which is the same instructions as the first. Again, not more than 200 words. And the third one, try to link these two together. So I would just read it. Discuss the link that the paper establishes between the 2 elements. Explain in what way the molecular element is linked to the neuronal or responsible to the neuronal behavioral element. And if the mechanism for this linkage is not yet known, if it's just an association, then if the mechanism for this link, if it is known, then describe it briefly. And if not, which is most usually the case, you just have an association. You know that mutation in the serotonin receptor, as we mentioned, can cause high chances to suffer from severe depression and so on. There are some ideas for mechanism in this instance, but nothing sound enough to, I think, to write in a paper. And so if the mechanism is known, then you definitely have to include it in your assignment. And if the mechanism is not known, then I don't know. You can just show what was the association that was established between the neuronal and the molecular and the neuronal element. Statistical
correlation between them? Or?
It's well, what were the experiments, for instance, what were the experiments that established a link between these 2 elements? How did they prove that such a link indeed exists in nature? Yeah. All in all, the assignment should not be more than 600 words. That's about 1 page. That's about 1 page. It's a very short assignment. Yeah. Okay.
Is it like, did you do that like that every year? Does it work? Because if I think of our biology assignments, I have the feeling like I write pages and pages and 600 words sounds very
Yeah, we do it every year. The thing is, when you write a paper, you have to start by write 3 pages if you want, and then go over it again and try to slash through the living flesh and just put out everything that is unnecessary. That's the way I work also. You write a paragraph and then you just cut it by half or a third or so on. Anything that's unnecessary just need to be left out. We do it every year. It works. Yeah, it works. It's okay.
Then is it has to be something like animals model, or you can take articles on humans, like?
Yeah, well, there's no restrictions on focusing on animal model, or you can use animal models, cellular models, humans, no models at all. Even bioinformatics that is only in what is called in silico experiments. No, there's no requirements about focusing on a specific animal model on it. Yeah. Any more questions about the assignment?
Should we print the paper when you hand in the assignment?
No. Okay, that's a good one. So just you send me the paper for an approval, and then you don't have to print it. It's a waste of page. I would just look at it online. So add the title of the article when you for the assignment. It's not written, but yeah, add the title of the authors of the article on the assignment. It will not be included in the 600 words. Yeah, that's that. Okay, so next time we will finish the molecular genetic techniques, and we'll go over the question page together.